Well, the Stem Cell Bioengineering Focus Group brings to bear technology on a wide variety of diseases. We're really focused on how you can use technology and quantitative science to understand questions in basic stem cell biology and in clinical translation. A number of the investigators use materials-based approaches to control stem cells. For example, we can culture cells on material spheres that can be suspended in culture to help us grow a large number of cells. Some of our investigators develop new chemicals to help regulate stem cell fate and differentiation. We're helping investigators bring genome editing so we can rewire the genetic makeup of cells. Some of our investigators look at imaging, so how can we look at a cell structure and help determine how effective it might be as a therapy, or can we track a cell inside of a body? A team of researchers in the Stem Cell Engineering Focus Group is making heart patches. So they make a material in which you can seed cells that were derived from stem cells, and these can then be placed on a patient who's had a heart attack. So this is an interface of the cardiovascular group with the Stem Cell Bioengineering group. We can help people in the neural group make specific brain type cells. We can integrate vasculature with those to make higher order and better models. One application of stem cells and the cells that are derived from them is looking at the toxicity of chemical compounds, trying to use stem cells as a way to predict which chemicals in our environment might be toxic to which cell types in our body. So researchers are taking stem cells and differentiating them to things like heart and liver and brain and blood vessel types of tissue, then exposing those cells to compounds that we routinely encounter but may not know what they do to the body. One area of emphasis of our focus group is being able to biomanufacture sufficient cells of the right safety and potency that they can be used in clinical trials. But it's difficult to make cells on the number scale that you might need to treat a patient. For example, if someone has had a heart attack, they lose about 1 billion cells in their heart. Now, we can easily grow 1,000 or even 1 million cells in the lab. So we help take lab scale processes, put these in a bioreactor, and grow enough cells to be able to treat a patient. It's also important that these cells have the right safety and potency profile. So what we try to do is determine what are the quality control and quality assurance tests that one might need to do on a cell before it can be put in a patient. What we try to do is provide the environment along with the cells. So we may inject the cells in a material that supports the cell's viability, allows us to control it while it's in the body, help it to stay alive and turn into the cell type we want, survive and flourish. Another challenge that we face is the ability to track what happens to a cell once we place it in the body. What we can often do there is genetically engineer the cell so it makes a fluorescent protein, and we can inject it with tracer particles that are magnetic, so we can use imaging-based methods to watch these cells. The University of Wisconsin-Madison is one of the few places in the world that has excellence in engineering, in medicine, and in basic biology. Students and other researchers who come here have exposure to cutting-edge science, medicine, technology. They can really put pieces together in a multidisciplinary way that allows them to get a great education and to make an impact on the world.